Mushroom Wonderland.
How's it going, everyone? Aaron Hilliard, Mushroom Wonderland, and in this video we're going to be learning a lot about trees and mycorrhizal networks and the importance of forest preservation. We were honored at Kitsap Peninsula Mycological Society to have Susan Simard from the University of British Columbia come and give us a talk about mother trees and the surrounding forest, and how these trees and the fungi and everything work together, how humans have played a role in helping these systems to work. It's just a super amazing talk. She was gracious enough to let us record this. We're just trying to get the information out there about how these mycorrhizal systems work in the forests, not only between fungi and trees, but between trees themselves, families of trees that live in the forest, and beautiful mother trees. So come along and watch this video as Susan Simard talks about how these forest systems work. She also is starred in a movie called Intelligent Trees, so check that out. It's just super interesting stuff about forest health, and we're all about that here at Mushroom Wonderland. Super interesting stuff. Thanks for hitting subscribe to Mushroom Wonderland, and try to support a forest near you. Let's get into this video.
It's my pleasure to be here, especially talking to mycophiles. I don't get to do that very often. And so I just want to start by saying, just to follow on and acknowledge that I am on the unceded territory of 3 nations, the First Nations of Aboriginal people here in Canada, the Sinixt, which overlaps with the American border, and recently this nation has been reinstated as a bona fide nation in Canada. They were declared extinct, actually, about, I don't know, 50 years ago, and we've just recognized them again as a nation in Canada, and they actually live, right through the territory. I'm in the Kootenay region. I'm also, it's overlapping with the territory of the Ktunaxa Nation, as well as the Okanagan Nation. And I just also wanted to acknowledge that my work has actually been in collaboration with many First Nations in British Columbia, and that's going to be infused through my talk today. I also just wanted to say that, I got my master's and PhD at Oregon State University, so not that far from where you guys are, and of course you have such a brain wealth in the United States about fungi and forests, and I've learned so much from my American colleagues and friends. And so I have to say that, a lot of this work was in collaboration with my American friends and mentors and teachers at Oregon State. I also just wanted to say, too, that I'm a forester. I was trained as a forester. I wasn't trained as a mycologist. But of course, I think anybody who wants to understand ecosystems needs to look at fungi. And so I come from it from a forest point of view. And of course, you'll see that in my lecture. And as you ask questions, you can, of course, ask me more detailed questions about the fungi, because my lecture is more about the ecosystem. So here we go. The book that I wrote is really, it's a journey, right? It's a memoir. It's a memoir that takes the science that I've done over the last 40 years, that I've published in many journal articles, and brings it to the public sphere. And I wanted to do this because I found that, people who are actually having big influences on forests where mushrooms grow, policymakers, practitioners really didn't know about it, or didn't understand it, or ignored this work. And I thought, we really need the public to understand this, to push our governments to make changes so that we protect our environments more carefully. And I've included here a spiral, because this is a spiral of a journey of my own, from when I was a child, where I grew up in forests, to where I am now, understanding the importance of connection of these relationships in forests for the whole global community and for our global biogeochemical cycles and society as a whole. So yes, Kirsten, I totally agree with you. This has got to be, I think, information brought to everybody's understanding. So I'm going to start with this map. You'll recognize this is British Columbia, just north of where you are. This is a map that actually shows the First Nations, the language groups of our First Nations in British Columbia. There's actually 200 nations that have been recognized, so this is just a simplified version of it. I live down here in this area right here, in the Ktunaxa, the Okanagan, and the Sinixt is this group that comes up, and they actually overlap with these two nations. But my work includes work in the Secwepemc, the Sekene, the Dakel, the Wet'suwet'en, the Silcotin, all the way with the Coast Salish, the Nuu-chah-nulth, the Kwakwaka'wakw, the Haltsik, the Tsimshian, the Haida nations. I work with all of these nations. And so their work, their understanding is brought to bear on this. And I guess, one thing I wanted to acknowledge too is that there was a Coast Salish man, Bruce Miller, or Subiye, who lived in Seattle, and he actually understood these connections that I'm going to talk about, these mycorrhizal networks, these mycelial networks that underlay the forest. This knowledge has been around for thousands of years, and it's really just, bringing Western science to uncover for the rest of us what has been known for a long, long time. So I just wanted to start with that. So this is where I grew up. I grew up in these forests. These are iconic inland rainforests. This is Western red cedar. The forests are mixed forests. They've got hemlock, they've got birch and aspen and larch and western white pine, lodgepole pine, at the high elevations, white bark pine. These are really important, iconic forests. They're very productive forests. So I grew up here. This is what I knew as whole ecosystems, as a kid playing in these environments and playing with my brother and sister and getting to know these at a very visceral level. And my family actually were loggers in these forests. They were horse loggers, and they lived along what's called the Shuswap River. And this is a salmon river. And I'm bringing this up because salmon influenced these forests all through the Pacific Northwest in a very deep way. The collapse of the salmon populations is tightly linked with how healthy our forests are, and of course how healthy all of the creatures in the forest, include the mycelium and the mycorrhiza. They're all interlinked together. And I'm going to come back to the importance of salmon and caring for salmon in forests later in my introduction in my presentation. So one of the things as a forester that you face is that even though I grew up in a province of old growth forests, it's turned into a province of clear cuts. This is devastating. It's devastating for me as a person. It's devastating for us as a people, for our First Nations people. It's devastating for our, for global change. This all contributes, it's all interlinked with it. And I study this. I understand the impact of this on our carbon cycle. I've studied the impact of this on our biodiversity below ground and above ground. The impacts are broad, they're extensive, they're long-term. And so it's this framework as a forester that I came in to study fungi and the importance of fungi and networks in forests. And as a young forester, I was dealing with the conversion of our old growth forests, which is all part of the plan, right? Of colonization came along with it, the plan to liquidate the old growth forests and turn them into second growth forests. So these are planted forests that are cultivated. They're planted with, often they're monocultures, and they get weeded out of diversity, really, the broadleaf trees, the trees and plants that are considered competitors with these trees. And the consequences of this are severe. I think that we didn't understand when we started doing this back in the 60s and 70s and 80s what the consequences would be. We didn't understand all the unexpected effects, but some of them are this. So, this is a plantation of lodgepole pine in the subboreal forest to the north of where I live. This has happened in many places where we convert these diverse forests into simple plantations. They get infested with beetles. They get infested with all kinds of insects. They get infested with pathogens. They're not very healthy. And I've studied this across British Columbia. We've found that, plantations that we thought were okay, 5 years after they're declared okay, we go back and we find that there are 50 damaging agents in these forests that have reduced density down by 50%. These are not healthy forests to be in. They're not healthy forests for the future. And so I wanted to understand, what were we doing? Like, we're basically dissecting our diverse forests and turning them into monocultures. And essentially what we're simplifying is not just the forest. We're simplifying the whole ecosystem below ground. And so when I went back to do my PhD, after learning about, forest practices, how we're simplifying forests, and I studied that, and I figured out that, these pine plantations weren't doing well. Why is it? What is it that we're doing wrong? And so I turned to the below ground community. I was heavily influenced by David Perry at Oregon State University and that whole team, Randy Molina, Jim Trappe, who were doing work on mycorrhizal fungi. And I wanted to go back. I thought that after my master's, I thought this is where we needed to look, was below ground. And so, these are the mycorrhizal fungi. They're one of 4 groups of fungi. You guys all know this. There's pathogens, there's saprotrophs, there's mycorrhizas, and then there's the endophytes. And I focused on the mycorrhizal fungi, the ones that I call the helper fungi, the ones that all of our trees are in an obligate relationship with in order to carry out the life cycle of trees and carry out the life cycle of the fungi. And I learned in the early 1980s about the amazing work that David Reed had done in the UK, where he had grown pine seedlings together in root boxes in the laboratory and inoculated them with a Psilithus, it was a Psilithus, and found that they could be connected together. And then he radioactively labeled one of these seedlings over here, and he traced that radioactivity right through this network, right into its neighboring seedling. And he also went so far as to shade these seedlings. He had the idea that this carbon was moving down source sink gradients, like phloem that goes from leaves to roots. They follow this source of carbohydrates down to sinks where those carbohydrates are used and metabolized. He had the idea that these networks served like phloem and xylem in a tree, and that carbon would move down these source sink gradients. So he went so far as to shade these little neighboring seedlings to different degrees, and he found that the more he shaded these seedlings, the more carbon actually moved over into this neighboring tree. That was amazing. That was incredible. He published that in Nature. And that was in 1983. I started doing my PhD in 1992, 9 years later. And really the work had kind of not gone that far. It was still in the lab. It was still stuck in the UK in these, in root boxes, or people were looking at them in grasslands and heathlands, but mostly not in big old forests like we're looking at in the Pacific Northwest, in Western North America. And so I wanted to see whether or not these networks existed in our forests, and if we were actually dismantling them through our very simple forest practices. And just to give you, this is just a picture that shows these mycelium that can actually link trees together. And now we know that these serve like pipelines, that the mycelium does have a complex structure, almost like xylem and phloem, where you can get this two-way movement going back and forth between one root and another, where carbon and nitrogen as amino acids and carbohydrates can actually move around the outside of a mycelium and through the core of the mycelium in 2 different directions, and that carbon and nitrogen and other compounds like phosphorus and water and anything dissolved in water can move back and forth between these roots. And so I set out to try to see this, to understand this in real forests, in our forests. I started my work in the interior dry belt forests of British Columbia. So these are Douglas fir forests. They're not unlike the coastal forests, except that the Douglas fir here is shade tolerant, whereas on the coast it's more shade intolerant. So when they're shade tolerant, that means that regeneration comes up in the understory of the old trees. So these trees here are about 250 to 300 years old, and you can see there's intermediate sized trees that are probably 50 or 100 years old, and then young trees coming up in the understory, the seedlings and saplings. And so this is an uneven age forest. It's highly structured, vertically structured, horizontally structured, and I started my work here after doing my initial work with my PhD in multi-species forests, I wanted to come back to these more simple forests, species simple above ground, to try and look at and map what these networks look like below ground in natural forests. So I got a graduate student. His name is Kevin Byler. I worked with Dan Durrell, who is another, great mycologist. I worked with Randy Molina, Dave Perry, Dave Myrold, all at Oregon State, and we set out to understand these forests and map what the networks look like below ground. So imagine this forest, and now I'm going to shift you over to a graphic of what that forest looks like from a below ground perspective. So imagine you're looking down on the forest, and each one of these circles represents a tree. And so the bigger and darker the circle is, the bigger and older the tree. So imagine this is a 300-year-old Douglas fir tree. And then these are all smaller trees of younger ages in this uneven age forest. And these little yellow ones are seedlings that are growing up in the network of these old trees. And so just imagine then these old trees, of course, they've got cones, they shed seed, the seed falls to the forest floor, those seedlings germinate, they establish, and they actually link into these networks. So these black lines represent actually the fungi that are linking trees together. So you can see this tree here is, and keeping in mind, so I'm sorry if I'm a little bit jumping around, but keeping in mind that we looked at in this forest where we knew that there were about 100 species of fungi per hectare, mycorrhizal fungi, we looked at two sister species of Rhizopogon vesiculosus and Rhizopogon vinicolor. And so this network that I'm showing you just represents those two sister species of Rhizopogon within that community of larger, of many different fungal species that also co-occurred. So if we had been able to map all of the species of fungi, this map would have looked much different. It would have looked more dense, more full. All the interstitial spaces would have been filled with mycelium. So let's look at the characteristics of this network. We analyzed it using graph theory, and we found all kinds of characteristics that are common to complex systems, to systems level thinking or any kind of system. So one of the things we noticed is that these big old trees are hubs in the network. They're the most highly connected. And this is a common feature of systems, whether it's a transportation system, a watershed system, a river system, a communication system. They all have these characteristics where there are complex systems have hubs in them. So they're the most highly connected. And this particular tree here is actually linked to 80% of the other trees in this forest. And these smaller trees, the seedlings, are less connected, but they are connected in the network of these older trees. And so, as I said, they germinate. Within 3 months, we found in our studies, they get colonized by the mycorrhizal fungi. They become part of this network. Other features of the network are that this is a resilient network. So, you see this tree here is linked to this tree here with multiple linkages, right? It's not just one fungus, one mycelium. It's many, many linkages. And this is what this multiple nodes of connection mean, that the system is very resilient. If a columbella comes in and eats off this fungus, or let's say it dies back and then regenerates, the network is still intact because there are many, many points of linkage. We started experimenting with these networks to see what they do. And around these old trees, we started by planting seedlings or seeds around them to see what they did. And what we found was that when seedlings can establish and join in the network of the old trees, that their survival goes up by between 25% and 4 times. And so it's a significant increase in survival. And why was that? Well, we found that these old trees are actually transmitting resources like carbon and nitrogen and water and phosphorus into these little seedlings. And later on, we found out they also transmit information. So if these old trees, for example, are suffering from some kind of herbivore like Western spruce budworm or Douglas fir bark beetle, that these trees will upregulate their own defense machinery, their RNA basically, and they start producing enzymes. These enzymes actually trigger a transmission of information from this tree, the the infected tree to the neighboring trees, and that they in turn, we found, will upregulate their own RNA and start producing enzymes that will protect them against the same insects and pathogens. This kind of work has been done in other places around the world, verifying this kind of communication, this information communication. And it's also been done in agricultural settings where people have found that this kind of defense messaging also goes on above ground. It's not just through mycorrhizal networks, it also happens through above ground pathways. So it's a very robust system, right? There's lots of pathways of communication going on here.
Hey, Suzanne.
Yes?
There's 2 questions in the chat about this slide, so I'll just read them to you now.
Oh, that sounds great. Yeah.
One, does the extent of the network shift with seasons? And secondly, how did you find the networks, and did you core the ground and look at the mycelia?
Okay, yeah, good. I don't know if it shifts with the seasons, but I bet it does. Of course, people who look at, you can use different techniques to look at seasonal changes in mycelium. We know that they do. The fungi fruit at different seasons, that the mycelium will grow through ingrowth mechanism using ingrowth tubes. We can see that the mycelium grows and more quickly and more abundantly at different times of the year, correlated with rainfall. And we've definitely looked at in earlier studies how mushrooms will come up at those rainy seasons. Around here, it's in June and in October. So yeah, I would think that they do vary with the seasons. The one thing that we did do is when we developed this network map in 2006, I had a student, Justine Carson, I had a student, and his name is Joseph Birch, and he went back about 15 years later and re-looked at this network. He went back into exactly the same forest, used this network map, and he found that not only do the seedlings respond to the network linkages, but so do the old trees. And we tried to remap the network, but we weren't very successful at it. But we were trying to see how it would have changed over time. But we expect, just based on looking at the trees, that there were shifts. There were shifts in emphasis of where carbon would go and what trees were responding. But I can't answer directly the seasonal question, except to infer from these studies that it would change over the seedlings. With respect to the methodology, what Kevin did is he went into this forest and sampled using a grid pattern. So he would sample around each tree in the cardinal directions for the fungi. So doing cores basically at the drip line of the trees. And then in between this tree, for example, in this tree, he did multiple cores in between them as well. And in those cores, he picked up whatever fungal material he could get, whether it was mycelium or spores or sclerotia, and did DNA analysis on them. And it wasn't just sequencing, because you can sequence and get species. What he did was what are called microsatellites. And so microsatellites can actually distinguish one individual genet from another individual genet. And we needed to do that in order to know whether this fungus that was on this tree was actually the same fungus that was on this tree. And we were able to do this only because in Randy Molina's lab, he'd worked with Annette Kretzer, and she had developed the microsatellites in order for us to actually, distinguish genets of Rhizopogon vinicolor and vesiculosis. And so it was work building on work of other people that allowed us to do this. We did the same thing with the trees. So he went in and he collected the cambium of the Douglas fir trees and used microsatellites developed in the lab of Sally Aiken at UBC. And we were able to distinguish the roots, for example. If he picked up a mycorrhizal root tip here, for example, not only could he identify the individual fungus, he could identify the individual tree that root was from based on the microsatellites of those Douglas fir individuals. So I hope that answers your question. There's a lot more detail in the journal articles, but yeah, it was very laborious. It was very time consuming. It took Kevin basically 5 years to do this study to make this map. Okay, so I'll move on. So one question led to another, and we kept doing studies. What were the importance of these old trees, and finding that they were important in the regenerative capacity of the forest. And so we started calling these old trees mother trees because they were basically nurturing the regeneration of the forest. This term mother tree, it's, I use the term because it helps people remember it to understand the role of regenerative role. But I have to say that, it's not really a scientific term. It's a term, Douglas fir it's got both sexes in a single tree. But I think that it conjures up in us an understanding. And the First Nations people, the Aboriginal people of North America, use terms like this in their languages as well. They called them grandmother trees or grandfather trees, mother trees and father trees, or elder trees. And so the language is important. It's important for us to understand and convey what we mean about the role of these trees in ecosystems, not just what they are, genetically. So thinking about the role as a regenerative part of the forest. The next question we asked, which I thought was a really logical question, was if these trees can actually facilitate the regeneration of the forest, can they recognize which ones are their own seedlings? Which ones of these regenerating trees come from these individual trees? Which ones are their own seeds? And this built on work that's being done with smaller plants, herbaceous plants, by others in other parts of the world. And I teamed up with one of those scientists. Her name is Dr. Susan Dudley. She's at McMaster University, and she started working on kin recognition in plants, about 20 years ago. And she worked with sea rocket plants, which are a clonal plant. And we got together and we said, well, I wonder, if all these trees are mycorrhizal, if they form networks, and we know that kin recognition, based on her work with her graduate students, happens through communication through roots. They knew that it was a root communication ability of these trees that allowed them to recognize their relatives. Could this be happening through mycorrhizal networks? And so we did a bunch of experiments, and she and I have had 3 graduate students working together and a postdoc who have dissected this. And we figured out with her that, yes, these trees can actually recognize which ones are their relatives. And the way we know this is because we've used carbon tracing, and we know that they send more carbon to relatives than to strangers. We know that the relative or the kin seedlings have bigger mycorrhizal networks. The fungi on them are more robust. They've got actually more fungal material on them. And also the seedlings grow faster. They've got more nutrition or different nutrition, and they survive better. And so what we call that is kin recognition, and the fact that they grow preferably in the neighborhood of their kin leads to this phenomenon called kin selection. That means that there's improved growth in the presence of neighbors or of relatives. Okay, so that's really cool. Mycorrhizas are involved in kin recognition in trees. Okay, I'm going to come back then to what First Nations have known all along. Like I said at the beginning, Subier talked about these fungal networks in the soil that the Coast Salish people knew about for a long, long time. And the Coast Salish people had a word for that connection. It's called "we are one, we are all together in this." And that word is Netsamotst. Netsamotst means we are all connected. This other word, Hishukishtawak, is actually a Nechonlith word. So those of you who have been keeping track of what's happening in Fairy Creek and Vancouver Island, the protests by the Nechonlith Nation and the environmental group, the protesters, have come up, talk about this word, about this phenomenon that we are all connected together. So this has been known. It's in the languages. It's not new. It's not new information to many people, but it's really cool to see this, that the mycorrhizas are very much of Hishuktishtuwak. Okay, I'm going to go back to the salmon forests. And I'm going to try and bring this story together with this last bit about, connection. So along the north coast in British Columbia, there have been many nations who have used this salmon fishing technology called tidal stone traps. And what we're looking at here is actually a satellite imagery looking down on a forest, obviously, and there's a river over here. And what the Heiltsuk Nation has done here, the Heiltsuk are at Bella Bella, is they've built these stone traps along the coastline. You can see the walls of stone here. And there are many miles and miles of these stone traps all along the coastline. And what they do is that when the salmon come in on the tide, when they're spawning, they come in on the ebb tide, on the tide. And when the tide goes out, the salmon are trapped behind these stone walls. And so the Haltzik Nation, the Tsimshian, whoever has built the walls, the Tlingit, they just passively collect the fish. It's a very simple fishing technology, but it's thousands of years old. So they collect the fish, but they always let the big mother fish go upstream and spawn. And so by doing this, what they were doing is they were actually manipulating the populations of the salmon. They were actually increasing the health of the salmon populations by favoring the big mother fish. And so keeping this in mind, over the thousands of years of this technology, we arrived, with colonization happened in North America, to these very robust salmon populations that were actually cultured and nurtured and managed by the many nations that lived along the Pacific coast. And so, we have images of this. We know that the First Nations depended on salmon, as they did with the cedar trees. They go hand in hand. Their livelihoods depended on fishing and drying salmon and using these passive fishing technologies. And those technologies vary depending a little bit whether you're inland or on the coast, but they were always these passive technologies that depended on always keeping the big fish so that they could build up the populations. This is one of the estuaries in the Haltzik Nation at a place called Hayat. And Hayat is an old 6,000-year-old fishing village that is being recovered. And in this village, they have tidal stone traps out here. You can't see them, but the salmon will spawn up this river. And they also have there are clam gardens here. There are trees that are cultivated here for cedar bark stripping, for making canoes. A lot of the villages along the coast also planted a number of trees. So you'll find crab apples and salmonberry, thimbleberry, blackberries. So you can actually, when you're approaching these villages, these ancient villages by boat, you can pick them out really easily because of the deciduous trees, the cultivated plants around them, the stone traps. So we started actually working in these rivers to find out what the influence of the salmon were on the forest. And so I have had a PhD student for the last 8 years actually working on this, as well as a Tsimshian Nation scientist, Dr. Teresa Ryan, who is also a salmon fisheries scientist. And we've been trying to building on the work of scientists at University of Washington, as well as Simon Fraser University, who have discovered that salmon nitrogen actually ends up in these trees. And we wanted to figure out the pathway. Were mycorrhizal networks involved in this? And so I'm not going to show you a lot of detail, except to say that the salmon have a deep, deep influence on the mycorrhizal networks. And now we know that, that the salmon, the density of salmon in these rivers, and we looked at 23 different rivers up the coast that had different numbers of salmon that were spawning in them. It was a natural salmon gradient, and we found that the mycorrhizal fungi varied very much along that gradient, that the salmon had a deep influence on the identity of the fungi in these forests. They also have a deep influence on the carbon sequestration of these forests and the fertility of the forests. These forests where salmon spawn have some of the highest carbon density of anywhere in the world. We haven't published that data, but we found that, and it's really profound. So what happens when the salmon go upriver is that grizzly bears and wolves and eagles will take the salmon that are spawning and they carry it into the forest. And they eat the salmon, especially under the big old trees. So they tend to go back to the same trees over and over again. They can see around them. They can see their predators. They can protect their cubs, and they eat the salmon under these old trees. And so the salmon, actually, the flesh of the salmon, which they don't eat most of it, they eat the brains and the guts, and they leave the most of the flesh there, and it decays and it ends up being taken up and acquired by these mycorrhizas and delivered into the tree, where we can actually take tree rings then and count the isotopic signature of salmon nitrogen in the tree rings and reconstruct salmon populations over time. It's really difficult to do. It's not easy, but we are kind of at the early stages of understanding at least what the effect of the salmon has on the fungal populations, the fungal communities, as well as carbon sequestration in these forests. And you can see the salmon bones in the forest. It's really cool when you go into these estuaries. It's not hard to find them. You just crawl along, follow the bear paths, which is what we did, these grizzly bear paths. And sure enough, it's almost predictable. You come up on these little benches and there are the bones. And there are many, many bones on these single sites just shining underneath these big old trees. So it's really cool. It shows the cycle, right? That everything is connected together, that the salmon foster big old trees like this, and they also shade the salmon streams, and they provide the detritus, the food web that actually feeds the salmon fry for the next generation of salmon to come. So it is a cycle. It is a linked thing. It's a network. It's a spiral. It all belongs together. And I think that's the end of my presentation. Yeah, I guess I took quite a bit of time, but yeah, that's my story for today, and I invite any questions that you have.
It's super awesome, Suzanne. It's super awesome. And yeah, you answered lots of questions which I would have asked you, but I see that there are lots of very interesting questions in our chat room. And maybe, John, we do it the other way around. You first answer the question of everybody, because I think that makes more sense. And then I can still add some if I haven't gotten it from somebody else.
Yeah, okay. I'll start with a question from Anita. Is there a relationship between moss and the mycorrhizal networks?
That's a great question. So as far as I know, mosses don't form mycorrhizas, they form rhizines. And the rhizines, the rhizines only penetrate so far into the soil or the bark or whatever substrate that they're working on or they're growing on. As far as I know, they don't form a mycorrhiza per se, but I haven't studied this recently. I had a graduate student, or actually working with an undergraduate student up at the Toolik Lake research camp way up in the Arctic, and we were trying to figure out whether or not the mosses were influenced by mycorrhizas, and we never actually ended up finishing that study. So I'm sorry that I can't answer it definitively, but as far as I know, they don't actually join into the network. But that doesn't mean they aren't part of the same system, right? The mosses, of course, will be linked in some other way. They moderate the temperature of the soil and the forest floor where the fungi grow, and of course they would add carbon and nitrogen. Some of them have nitrogen fixing bacteria associated with them, and that will also be important to the fertility of the forest and the tree growth, and therefore the mycorrhizal fungi that form these connections. So I don't know about the direct link, but I know that indirectly they are all linked. They are all in this interacting with each other.
Okay. Next question. Does the novel The Overstory represent the science of your work accurately and fairly?
I love the Overstory. I thought it was a wonderful book. I think that that character Patricia Westerford, from what I understand from listening to Richard Powers' interviews, is a combination of me as well as a couple of other people. So it's not just me. And I think that character reflects that. The research I did was on mycorrhizas and fungal connections. And what Patricia was actually studying was communication above ground. And so, I mentioned that other researchers were actually working on this, that trees will warn each other through emitting volatile organic compounds that other trees detect through the air, which is a more general way of communication than the more specific avenues through mycorrhizas. And so in some ways, she did represent me, but in the details, no, not really, not the details of my research. And of course, Patricia also was faced with a number of personal adversities in her career, and I think that in the end, she didn't actually make it through. Right? She did get her voice back, but not in a really powerful way. And I'm hoping that I can get a little bit further than Patricia did.
Yes. Can I chime into this? So, is there a sort of interaction or talking between trees over the air?
Well, yeah, there are. So these volatile organic compounds, we all smell them when we're walking through the forest. We all smell these organic compounds when we breathe in the scents, right? And those are communication molecules. And so what people have done is especially in shrubs and herbaceous plants, is they've purposely injured these plants and then measured these VOCs that transmit through the air, and the neighbors pick them up. And so, yeah, it does happen. It's been well verified. It's been studied a lot. And it's been known for, I don't know, probably 20 or 30 years now.
Okay.
Before we knew about the mycorrhizal connection and that communication pathway.
Yeah. Okay.
Okay, a question from Rachel Friedman, who did her undergraduate research with Jim Trapp.
Okay.
And she's curious about your use of the term mycorrhizas. And can you help understand the difference between that and the multiple mycorrhizae?
Mycorrhizae and mycorrhizas?
Yes.
I think that the mycorrhizas is more of a European use of the word, and mycorrhizae is more North American. That's how I've figured it out. They both mean the same thing. It's just the plural of a mycorrhiza. They're interchangeable.
Yeah.
Okay. All right. Got to process one other question. Let's see. Does your research explain why some species, like chanterelles, often fruit only when the trees are above a certain age?
I wish I knew the answer to that question. I knew that you guys are going to ask me questions that I couldn't answer, but I would expect so, but I don't know for sure. I haven't studied that. But, think about, chanterelles, as all mycelium, all mushrooms depend on carbohydrates from the trees. They don't survive without them, except as maybe resting spores and sclerotia. And so the more carbohydrates that are fixed through photosynthesis by the trees, the more carbon ends up below ground. It's just, and it depends on the ecosystem you're in, but anywhere between 20 and 80% of photosynthate ends up below ground in the soil food web and in the mycelium. And in more drier or poorer ecosystems, the more carbon goes down there. In more replete ecosystems, like, for example, coastal rainforests, that number will go down. There'll be less allocated below ground because trees can get their resources, they're more plentiful, so they can get them more easily. What we found in our studies in the Mother Tree Project is that about half of the carbon in our Douglas fir forest ends up below ground. It doesn't really matter. There's a slight response from dry to wet where there's a little bit more allocated below ground in drier forests compared to wetter forests, but it's pretty much a 50-50 thing. But, so getting back to the question, the more carbohydrate that ends up in the mycelium, the more that will be available for fruiting. And so I think, without studying it, that's a general principle that you could apply to understanding chanterelle fruiting. But, that's as far as I can shed light on that, as far as I know.
Do you have any data or just your personal observations? Like if you have a clear cut and then the clear cut regrows either from alone naturally or because they plant trees or whatever, at what point, so after how many years will you find fruiting of the mushrooms which were there before, which don't show up as a clear cut? I mean, this isn't like, a desert kind of thing. Doesn't look very good. There's nothing popping up. So that would be definitely a thing which mushroom people would want to know, because some of our nice, hunting grounds got wasted,
Yeah. I know. So we did a chronosequence in the mixed Douglas fir birch forest in the interior BC, and we got data on this. So we actually only followed from a clear cut to 100-year-old forest, but we got clear cut 5, 20, 50, 100-year-old forests and a few in between, well replicated study. And we found that and we looked at the DNA of the mycorrhizal fungi below ground. We also looked at the mushrooms. Looking at the below ground mycelium and the root tips gave us a clearer picture, because it's hard to follow mushrooms, but because they vary so much from year to year, as you all know. But what we found is that in the first few years, the first 5 to 10 years after clear cutting, you go from a forest that has about 100 species per hectare of fungal species, mycorrhizal fungi in general, on average, to about 4 or 5. And those 4 or 5 are, don't require a lot of carbon. They're things like Thelephora and Wilcoxina, Pizzalis. They're really, really simple fungi. And then, and low carbon demanding fungi. And then over time, the fungal community starts to recover, and Rhizopogon starts to come on maybe at 10 years. But the full suite, the diversity of that community doesn't really recover until the forest is at least 50 years old. And so that's when I would say you start to see a fuller community of the fruiting bodies, and then it just grows from there. It kind of starts to level off. And so by a century, you're almost fully recovered from what we can tell. And then, as the forest gets older and older, of course, there are more fungi, more rare fungi are added onto that diversity list, that richness list. And so, even 100-year-old forest is not going to fully represent what was there naturally in an old growth forest. But so just to summarize, about 50 years is when we start to see a fuller recovery, almost a full recovery of the community.
Yeah. And that is super sad for me, because I won't be alive then anymore.
Yeah, I know. And, of course, you see some things like morels come up following fire. And, it's funny, I've got 2 daughters, and they've become both in forestry. And one of my daughters was working up north of Prince George, and there were huge fires when she was up there, and large clear cuts, and they've clear cut like so much, but instead of forestry now, it's basically mushroom pickers that are up there, and there are huge camps of mushroom pickers. It's unregulated for the most part, and they're picking morels, and they're selling like it's a huge market. And in fire years, that community is massive. And so, there's a lot of mushrooms after clear cutting, but they're mostly morels.
Yeah, which is in some ways also nice. But if everything just looks like a desert, that doesn't, make any sense. It's not good for anything. So,
No.
Yeah. And even if you love morels, I do think that most of us would agree that cannot be the goal, that we just burn everything down to get morels. Yeah.
Yeah, no, definitely. And that definitely was never the goal. It was just like they didn't care at all about mushrooms. It's just that they popped up. And so then the whole community of this sort of underground community of unregulated, but I don't know. Yeah. People that are trying to make money from the land.
Yeah. Well, taking the opportunity because it's there, and it tastes really good. But since we talked about fires, since you mentioned forest fires, what's your take on them? We have more and more coming because of the climate change and whatever. Do you think we have to change, or the forest industry has to change something? Well, what would you recommend? What should be done?
Yeah, it's a complex problem. It's a wicked problem. We've had a huge fire year up here this year in British Columbia. In my little town of Nelson, in July and August, I was surrounded by 5 huge fires. We couldn't even breathe for like a month. People were migrating from the interior to the coast just to breathe. And this is going to, last year wasn't so bad, but the year before and the year before that, it was again a disaster. We had like millions of hectares burning. This is the future. The future is now. It's here. There's so much we can do to mitigate this. So, for one, one of the things that we've found, or people are talking about this year that we've noticed, and and I've been talking about this for a long time, as of other people, but how we how we clear cut and then replace old forests with Douglas fir, lodgepole pine, spruce, simplified monocultures, and then weed out the broadleaf trees. We're actually increasing the flammability of the landscape. So not just the individual clear cuts are highly flammable, but the pattern of clear cuts, that there's one after the other, means that the fire, once it starts, it just rages through the landscape. And then without big old trees there, which have thick bark and deeper root systems, which actually are resistant to fire, at least in the interior part of the province, when they're there, they help the forest recover. Obviously, they provide seed, but the roots actually will bring water up from down below and share it through the mycorrhizal network with the other plants and trees and keep the forest moist. And of course, transpiration also keeps the forest moist. There's so many things that do that. And the fires that are burning through these clear cuts are actually drying out the landscape. So what can we do? We can leave big old trees, even when we harvest, like do partial cutting and leave old trees. They'll host the mycorrhizal fungi, and they'll also, help the landscape resist fire. So there's resilience and resistance built into leaving old trees, and then not planting them to monocultures of flammable trees, like having mixes of broadleaves and conifers of all these species also will really help. And of course, on top of that, we need to reduce our rate of cutting so that we don't have a sea of land of clear cuts out there. We need to address our fossil fuel consumption so that we can decarbonize our energy sector. We need to address this heating of our atmosphere so we don't have fires that are being created by other fires. So essentially, it's become so hot and dry that when we get a fire, it creates lightning itself, which spawns even more fires, which sounds like a disaster, and it is, but we do have tools, right? We do know that if we leave old trees and don't create these vulnerable clear cuts, plantations, that we can mitigate the spread of fire. There are things we can do.
Right. There's lots more questions from the audience.
Yeah.
I'll move on to the next one. This is from Pete Allen, and it's a question that I had prepared also. But essentially, it comes down to, we can see trees all the time. We can rarely see mushrooms. More, I guess, if you're looking for them, but they're not so obvious. And so, as we look at this relationship, like, who's in charge? Is it the trees that are kind of telling the mushrooms what to do? Or are the mushrooms telling the trees what to do? Or is it truly a symbiotic network? Or does anybody know?
What do you think?
Oh, it's definitely the mushrooms that are in charge.
Yeah, we're mushroom nerds here. That's
well, yeah. I would say they're both involved. All of them are involved. There isn't anybody in charge. It's a complex system. It's about the relationships between all of them. As a forester, I studied how trees regulate transmission of carbon back and forth through mycorrhizas. And everything I did, I manipulated these source sink gradients by shading, by using nutrients, by using varying species, by varying the water capacity of the soil. Everything mattered. Right? It all influenced what went where. So definitely what the status of the trees is important, whether they're big or small or rich or poor, that all matters. So does the mycorrhizal networks. The identity of fungi in those networks also matters. Whether it's, full of Wilcoxina or full of Rhizopogon, that matters. And so who's in charge? Well, they're all working together. It's about their relationships. And, we have to, try to stop ourselves from trying to think simply about the system and say it's one or the other. We need all of these things together.
Nice. Okay.
And of course, the mycorrhizal people study the mycorrhizal. I mean, if you look at one thing, you'll find it. And, the challenge is look at all these things together, right? That's really hard to do. That's where the next studies need to go.
Yes. Who is, like, what's the title for somebody who's doing that? Because the foresters aren't necessarily doing it, and the mushroom people aren't doing it. Like, who's doing it? Like the ecologists?
Well, yeah, I mean, I'm a forester. I'm a forest ecologist. I guess the ecologists, they're the ones that look at relationships between different species. So yeah, it's on them. But really, it's it was really the mycophiles who started this. It wasn't the tree people who started looking at this. It's you guys that spawned the questions, right? And then the rest of us are trying to understand the ecosystem, say, oh, these mycophiles, these mycorrhizae people, they know something about the below ground world. Maybe I should look there. And so then we start looking at the relationship between our trees and your fungi, and we find out that they're both important. So, I think it's people getting together and doing the work together is really important to this collaboration. And I have to say that, as I started out in government, and now I'm an academic. Academia is not the place to do collaborative work, unfortunately. Academia needs to be completely restructured so that it honors and fosters collaborative work. It doesn't do that. So it's really up to us as individuals to seek out those collaborations and try to do them. Work with scientists and naturalists and government people and academic people, everybody together, as much as we can.
Okay, lots more questions, but I'm going to ask this, even though nobody brought it up, even though I know that I want to know. Do you still play goalie and drink beer?
I'm thinking about my beer right now.
Okay. So, what would you hope that your work would inspire in a group of amateur mycologists like KPMS with 200 plus membership families to do going forward?
I think well, education is huge. So talk to your friends, spread the word, get the education material out there, get people out in the forest to look at these creatures, right? Because once you get people out there, they fall in love with it right away. You probably all know this already. Get people so they're hooked into looking for mushrooms. And then, so that's number one. The second thing, and maybe this is most important, is that we need to protect our forests. Like without forests, we don't have any fungi, we don't have mushrooms, and our forests are disappearing really, really rapidly. I mean, I think that in the US, you've made some progress by, in some ways by, there's new forestry that Jerry Franklin started, and that started with, the Pacific Northwest Forest Plan, which protected some forest. Up in Canada, we're still clear cutting as though, our lives depend on it, when our lives actually depend on saving these forests. And we have a lot to learn. And so I think that a lot of energy has got to be going into protecting the forests, saving the old growth that we have, as much as we can. Like it's dire, it's essential. So I spend a lot of time actually supporting people who are out protesting against logging. I support, I'm more and more, as we see our forests disappearing so rapidly, this is where we need to put our energy in. Without the trees, we don't have the fungi.
Yeah.
Excellent. Another question from Bob Johnson. You mentioned the salmon influence on the forest, and can you talk a little bit about how does the forest influence the estuary and the ocean, and what are the connections between the forest and the sea? I mean,
okay, yeah. So of course they're linked. The ocean forest interface is a very active place. Lots of people live there because there's a lot of activity. There's a lot of food. There's a lot of species there. All ecozones have those kind of features. Salmon are the vector that go between the forest and the ocean. And so, if the salmon are being overfished in the ocean, that means there's less salmon going into the forest and less salmon that is fertilizing the forest, the mycorrhizas, the networks, fewer bears are able to survive, fewer eagles, fewer wolves. So it trickles all the way up the food chain and all the way down the food chain. And when the forests are less fertile because there's fewer salmon, that means that the rivers are also less fertile. And especially, if we're clear cutting these forests as well, which we're doing as fast as we possibly can, then the food web of the streams is disrupted, because the food web of the streams is driven by the litter fall that's coming from those trees and from the roots and the euphoric zone, and where water moves back and forth from the tree to the stream in these underground channels. And so, if the trees are not there or they're less productive, that translates automatically into the fertility of the streams, which affects the ability of the salmon to spawn and the fry to grow and then go back out to the ocean. So overfishing, over harvesting, those things interact, and we've seen the decline in the salmon populations, actually the crashing of the sockeye salmon populations, and this is going to affect the forest. And I'm going to actually, because I don't know a lot of detail about it, but what we are doing, this is an interesting study. My research associate, Teresa Ryan, who is a Tsimshian woman, who's a salmon fisheries scientist, what we're doing to try and get a handle on this question is when colonization happened in North America, in Canada, what happened was that the Confederation is that the government basically tried to destroy the Indians. They put the children in residential schools, and they also, we started logging our forests and we started fishing the oceans. And basically, the Department of Fisheries and Oceans in Canada took the resource, the salmon resource, away from the First Nations. They basically plowed in those tidal stone traps and said, you can't use them anymore. We're taking over the fishery from here. And that happened in the early 1900s. And so then they said, okay, we're going to manage the fishery. Well, we know what happened from there. The salmon populations crashed. We overfished the salmon. We didn't fish on the tidal cycles. We didn't passively fish. We harvested everything. And so now what we're doing, Teresa and I, is we're going back to these old tidal stone traps and we're reconstructing some of them. And we're doing them in some of these really rich salmon rivers, and we're going to track how that, and we're going to, in reinstating and revitalizing these stone traps and using the old fishing technologies, we're going to trace, using nitrogen tracers, how that's affecting the nitrogen productivity of those estuaries, and we're going to be tracing how that affects the productivity and the nitrogen capacity of those forests. And so we're trying to answer that question. It's not easy. Doing any kind of work with Aboriginal people means that there's all kinds of history to grapple with, and even going even with the First Nations people who are collaborating with you. These are archaeological sites, right? These tidal stone traps are thousands of years old. It's like going and doing an archaeological dig for a dinosaur, right? It's that historical. And so it's taken us 3 years just to get the permits to go in there and start doing our work. So yeah, it's painful work, it's difficult work, it's laborious work, it's going to take a long time, but we have our hypothesis, which I just laid out for you. We think that these things are intimately interlinked, and we're going to try and verify that with our measurements.
We probably need more from the First Nation young people who study that and want to do it themselves, because then it's their land, it's their heritage, and then their interest is the biggest to keep it, or better to fix it up again.
It's exactly. And, we're going to be working with the Haltzik youth with these 3 stone traps. They're going to be rebuilding the stone traps. And I just wanted to, just say one little quote from a Haltzik woman, Jess Husty, and she's a mother, and she lives in Bella Bella. She's a scientist as well, and she's been trained as a Western scientist as well as she's an Aboriginal scientist, and she talks about how Western science is the little sister of Aboriginal science, and really we have a lot of learning working with them to learn about these systems, to learn about this worldview, to change our own worldview so that we can revitalize our own world, and move into a more productive society.
Yep. It's
Two,
almost like Robin Wall Kimmerer's braiding
Yes.
dress thing. Yeah.
two very specific questions
Exactly.
from Sue. Did your investigations find any mycorrhiza interface with rhododendrons? And secondly, how does soil type influence the distribution of the mycorrhizal relationships and abundance?
Yeah, so I don't actually look at these ericoid mycorrhizas myself, but one of my PhD students did for her master's research, and Shannon Birch, who is actually a trained mycologist from Oregon State and works in our government in Canada now. I think she might be retired now. Did a lot of work with ericoid mycorrhizas and rhododendron and salal, especially. And yeah. So what specifically was the question? Sorry, I don't know a lot about them. I mean, I know that these are very specialized mycorrhizal fungi. They're only certain species, a low number of species. They're not just mycorrhizas. They also can decompose organic material so that these plants can uptake organic nutrients. They're essential in the productivity of these forests, but I don't study them myself. Was there another part to that question?
A soil type on mycorrhizal.
Yeah. So I would say that soil type, what does that mean, right? In Canada, we have all these soil orders. They're genetic soil orders. I know that you have soils that are developed in the same way. We have glaciated soils. Most of the American soils are not glaciated. So that makes a difference whether they're glaciated or not. Glaciated soils tend to be poorer soils. They're younger soils. They're less weathered. And so the mycorrhizas that form on those soil types, I think are they're good at extracting nutrients from difficult situations from poor soils. And also as you move north, we get into more boreal forests, and the mycorrhizas and the plant community changes. And so those soils are even younger, and they're even more nutrient poor. And there's a lot more ericoid mycorrhizas in those communities. And then as you move southward and go into the tropical forest, you move from like more ericoid to ecto in these temperate forests to more arbuscular mycorrhizal dominated communities in tropical forests. That's very general, of course, but that tells you that those associated with those changes are changes in soil type as well. So you'll go from these deep organic soils in the boreal forest to podzolic soils, or spotosols as you call them in the US, in the temperate forests, to more septisols or luvisols and brunosols. That's what we call them in our temperate forests, down to lateritic soils in the tropical forests. And so the mycorrhizals change along that gradient too. So what drives that? What is it? Is it soil or climate? I mean, they all do work together, but climate, I would say, is the number one factor that affects soil weathering, soil development, and then the plant community that's on them, and that plant community is associated with a certain mycorrhizal community. But to sort out the hierarchy of these things, I think climate is the number one thing, soil is the next thing, and then the plant community is the thing next after that, if we were to put them in a hierarchy of importance.
Awesome.
Can I pop in a thing? With the clear cuts, we don't change the soil, but we change the plants which wander in, like Scotch broom or like ivy. Do those things change the mycorrhizae then also? Because you could argue the mycorrhizae are underground, they sit there and they wait until things grow back and then they do more action, but maybe not because they don't have any host trees. What is that? Yeah,
I mean, a lot of those weedy plants, like I think Scotch broom is an ericaceous plant. Ivy is it's either arbuscular mycorrhizal, or maybe some of these weedy plants are non-mycorrhizal. A lot of weedy plants, are either non-mycorrhizal or arbuscular mycorrhizal, whereas our forests are generally more ectomycorrhizal, at least here in the Northern Hemisphere. And so it's a wholesale shift from a whole functional group of mycorrhizas to another functional group of mycorrhizas. So of course that's going to influence, the productivity of that site, as well as the ability of the plants to uptake nutrients from the soil. It'll have a profound effect. And here's one story I can tell. When I was a younger researcher in the government, I did all these crazy experiments. And actually, I talk about one of them in the book, where I actually made a big mistake and I destroyed this site. I actually, in my zeal to create this perfect experiment, I got rid of the forest floor and all these weeds moved in. And the mycorrhizal community completely changed from ectomycorrhizal to arbuscular mycorrhizal. I couldn't get conifers to come back. I kept planting over and over and over again. It was like I created this black hole. And it's because the whole mycorrhizal community had changed. And it was only when I was able to transfer soil back from the forest into that clear cut could I regenerate the forest. Yeah. So how we change the plant community changes the mycorrhizas, which really affects, the recovery of that plant community.
Mm-hmm. Yeah.
Another question from John Blendauer. How does forest thinning affect the health of the remaining trees in the short term? And how does the removal of the large mother trees versus thinning out the overcrowded smaller trees affect the eventual rebound of the same forest?
Yeah, I think, these big old trees, they have the big crowns. They're highly photosynthetic. They support, a kind of a more diverse community of mycorrhizal fungi. Of more, the older the tree, the more of these kind of old growth or multi-stage fungi you get on them, versus younger, smaller trees that don't have that photosynthetic capacity, can't support the same diverse community as the old trees. And so, really, and these younger trees that sort of in Douglas fir forests, especially where we do a lot of fire suppression, we get a lot of these young trees growing up under the understory of the old trees, and they kind of sap these old trees of their vitality. They compete for water. They take up water. They take up space. I think that if we're going to be restoring these communities, it's to keep the old trees and, get rid of the understory through prescribed burning, and that will revitalize the forest and it will help the mycorrhizal community become more diverse. So, it's not going to be the same prescription everywhere, but I think in general, and looking at our forests up in Canada and these dry belt forests where this has happened with fire suppression, this is what we need to do. We really need to restore prescribed burning, as the Aboriginal people did a long time ago, to bring back the resistance, the resilience of these forests and increase the diversity of the below ground community.
Great. Let's see. Going down. Okay. This is from Aaron. Good question. Humans always get a bad rap. Are there any obvious positive roles that humans play amongst the forest networks?
Totally. I think that the Aboriginal example of the salmon is a great one, where they have this reciprocal relationship between the salmon, the forest, and their own communities. Right. And we as human beings, we can cultivate really robust communities. We're geniuses at this. We also get led astray, and where it can be exploitive. But if we can get back to more this productive role, we can have a hugely positive influence. And one of the things that we really need to do as climate is changing is that, the velocity of climate change is so much faster than trees can adapt or migrate, that it's going to be our job to help trees move. We're going to have to be planting genotypes, in different climates in order for these trees to stay healthy and alive, and to keep the forest vital, and to ability to keep sequestering and storing carbon. If we don't do that, we're hooped. We have to do that, and we can do that. We've studied it. There are good scientists out there who understand that. But when we move these trees, we need to move them into receptive communities. So it can't be like into, clear cuts that are devoid of other plants and mycorrhizal fungi and bacteria and the soil food web. They have to go into healthy environments. And so that means that, retaining these old trees and then migrating genotypes into these always intact networks. We're going to have to play a role in that. We have to play a very productive role in that.
Okay. Some of the invasive and non-native species are allelopathic for plants. Is there something similar that's true for fungi?
That fungi can be allelopathic, you mean, for other fungi? Or well, let me take this a little bit. So one of the things, so a lot of noxious weeds can be allelopathic. So that means that they can produce chemicals that prohibit or inhibit other plants from establishing around them. There's even native trees and plants that do that. There's the famous example of the black walnut, which produces a compound called juglans, which inhibits other plants from growing in the understory of black walnut trees. But there's one story I'm thinking of, and that's with knapweed, where knapweed is a very noxious weed. It's very invasive. It can even invade into intact grasslands and intact forests, even when there's no disturbance there. And one of the ways that, and this was scientists actually in Montana that looked at this, that they do this is that they have arbuscular mycorrhizal networks. And these arbuscular mycorrhizal networks actually tap into the arbuscular networks of the native grasslands. And the knapweed will steal the phosphorus right out of those grasses. And it kills the grasses and allows them to invade into those intact communities. And so in a sense, the fungi are working in concert with the knapweed to invade and be, and it's kind of is it allelopathic? Is it poisonous? Well, certainly a knapweed community looks pretty poisonous to me. There's not much that can grow around them. So I think that there are examples of that, but I'm not an expert in this area at all. So yeah.
Okay, another question. You talk in your book about your research and your methods about using Western red cedars that form arbuscular mycorrhiza as a control. And then so currently is it thought that arbuscular mycorrhizal networks are completely independent from ectomycorrhizal networks, or do some fungi and plants participate in both?
Yeah, there are. So there's some tree species and shrub species that form both arbuscular and ectomycorrhizas. So willows, for example, in the Populus genus, eucalyptus is another one. So there's not very many, but there are some. And also we know that this was done in Dave Perry's lab, where you grow, for example, Douglas fir in a grassland full of arbuscular mycorrhizas, that Douglas fir can form what are putative arbuscular mycorrhizas when that's absent of other ectomycorrhizal inoculum. And so the neighborhood of the tree matters, and the community that is trying to grow in matters. They can actually form these kind of weird mycorrhizal associations, what they don't normally do. In the ericoid ectomycorrhizal association, there are fungi that can form an ericoid mycorrhiza on an Ericaceae plant and an ectomycorrhizal on an ectomycorrhizal tree and actually join them together. So that's kind of weird, but cool. So those are just a few stories where they can be quite flexible, and it depends on the environment that they're in the plant community. Context matters.
That's amazing.
Kirsten, what else you got?
I see so many other questions. You really want me to do it?
No, I'm just trying to copy all those down. So if you can,
if I can switch one in, okay. Well, I was reading in your book mainly about, or what kind of popped out for me was this one thing. You mentioned Darwin and his words and his works, and that he is kind of always only interpreted as a, survival of the fittest and just competition. Mm-hmm. And you put out this completely different looking at this, that he is also talking about working together in collaboration. Mm-hmm. Almost reciprocity. And so how do you think we can change our thinking from this? Because this is like in not only the forest industry, this is also like in agriculture, pretty much everywhere we humans work, we think it has to be all competition, but it should be more collaboration. So how do you think we change this? Because we need to change it in order to survive, I guess.
Yeah. Competition, collaboration, they happen all at once, all at the same time, right? So you can have, two trees side by side that are collaborating and competing together.
Yeah.
So it's simultaneous, it's complex. So to say it's one or the other is false too. So it's multifarious. And how do we change our mindset? I think that scientifically we've made the shift. I think that even, we now know that, the evolution of eukaryotic cells was really a symbiosis from the very beginning. We know from the Human Genome Project that we're full of viral DNA and bacterial DNA. We're not just like individuals.
Yeah.
And so we've made that shift. We have that understanding. It's the translation of that knowledge into practice that's the difficult part. So we've got this huge, as you say, infrastructure in forestry and agriculture, aquaculture as well, where we've designed all these practices on this basis that we need to get rid of competitors or to simplify these systems. And so we really need, it needs to be like, I guess we need to be advocate, or we need to really educate people and advocate for these changes, because it's killing us to be so simple. We need to become more sophisticated in what we do, and what we're fighting against is this built up infrastructure around this idea of competition, right? The herbicide companies, the genetic companies, the fertilizer companies, it's a huge, big business, and they're going to be resistant to change. But we've got to, I guess at the grassroots, start making these influences. We've got to, make these changes, and we can't linger. We've got to do it. We've got to do it quickly.
Maybe even change the laws, because we cannot sue on behalf of a tree. We can sue on behalf of a ship.
Yes.
Yeah, we cannot sue on behalf of a river, of a forest who is taken away. Like the First Nations, can they sue us, the incoming people? We mess it up. So there is no
It's not easy. Yeah. It's not easy, but our health is being impacted by these kind of backward policies, like the fires, for example. We can if I can't breathe, and if I die or my kids die, or and people are dropping dead from this, like in Vancouver during the heat wave, there were 800 people that dropped dead in about 2 days just from heat exhaustion. And so it is affecting our human health. And so then when we humans, we can advocate for our own selves, but it is really about conservation of our natural ecosystem. So I don't think it's a hopeless situation, but we do need more direct avenues to put value on our health, our public health, our public ecosystems. The laws do need to change. And I think that I'm actually working with some lawyers right now in trying to sue the government of Canada, of British Columbia, because, they're going to cut down Fairy Creek. And Fairy Creek is like our last iconic old growth forest left on Vancouver Island. And it's a huge storehouse for carbon and biodiversity. And what that lawsuit is trying to do is change the law so that you can actually sue on the behalf of the public health, not just on the individual actions of the police, for example, or the forest company.
Yeah, that's awesome. That's cool. So maybe we should go up there and climb in trees and live there so that they can cut it down.
That's what people are doing. Honestly, tomorrow there's going to be a huge protest of elders, people that are, in their retiring years, and they're going to march up that Fairy Creek Road to stand with the young protesters. It is all of us, and people are putting their lives on the line. Like there was an elder woman, I'm just going to give this example, who drove her car to Fairy Creek yesterday to stand with the young protesters, these 20 and 30-year-old kids. And she was pulled out of her car by the police And then the forest company drove their skidder over top of the car and crushed it.
Wow.
I mean, it's like, it's like the Taliban, for Pete's sakes, right? Like, what are we doing? It's crazy, and we need to change these. We need to change this. People are trying to stand up for the health of their kids, for themselves, for their families, and the governments are not, and the industries are not responding, and they need to respond.
Yeah.
And people are going to dramatic ends to make them respond. And I think it's going to change, but it's going to take a lot of pain. But we have to keep pushing.
A lot of people doing. Yeah.
Yeah.
Huh. That's cool.
Okay,
All right, John.
One more question from Drew Haven. Do you have any advice for aspiring science communicators? I'm reading your book right now, and I'm so interested in the process of integrating the research and results into a narrative that is broadly accessible.
Yeah, like I wrote in my book, I never really thought of myself as a communicator, but over time, as I've gained more knowledge and confidence, I've become something like that. And it really is just, take your heart and put it out there and stand up for what you believe in it. And if you do that, people will listen to you. You don't need special skills. You just need to practice doing it. And that's all I did. But if you're more, if you can get some training and become more outgoing, if you're introverted like I am, make yourself do it and practice it over and over again, then you can start to have an influence, and not just using one mode of communication, but many modes of communication. So, I started out, giving talks, and then I started teaching, and then I started doing videos, and then I did a TED Talk, and then I wrote a book. And it was kind of this step-by-step process for me. But when you're younger, if you could start out earlier saying, this is what I want to do, you'll be way more effective, right? Than sitting in your scientific lab and doing, taking decades to get to that point. I think it's really important that people do speak out, that you learn how to do that, to speak up for the forest, because it really needs us to do that.
All right. There are many more questions. We had a great turnout tonight for your presentation, Suzanne. So thank you so much for being here. But I think 90 minutes is more than we were hoping to get. So
hold on, John. Hold on. One more thing.
Oh yeah, what's that?
The Mother Tree Project. Can you explain a little bit about that? And because that's another really cool way of that people can get involved. So I'd like to talk. Carmen,
that's a great question, and I'm happy to answer that question. So the Mother Tree Project is a project I started about 6 years ago, and the objective is to how do we manage our forests so that they remain resilient as climate changes? That's the overall question. And I started it from my understanding of how mycorrhizal networks work in forests. And so the idea is, how do we conserve these networks? And keeping in mind that mycorrhizas are a symbol of all the connections in forests. There's other microbial networks, there's bird networks, there's stream networks, there's all kinds of networks, all these connections. How do we conserve these? And keeping in mind that trees are the photosynthetic machinery that feeds these ecosystems. That's where the energy comes from. So we need to conserve these old trees, and we can do this in different ways. And so we started playing around with, versus clear cutting. What's an alternative to clear cutting? Well, we can leave individual trees, we can leave groups of trees, we can leave large contiguous patches of trees. We can leave the forest intact. And so we're comparing all of these kinds of ways of leaving old trees and their networks intact across different climatic, a climatic gradient that goes from the hot, dry communities near the 49th parallel all the way up to cold, wet communities in the subboreal forests. We have forests now that we're continuing this network up into the coastal ecosystems as well. And we've harvested them in different ways in collaboration with community forests, First Nations forests, industrial forests, research forests. We have tons of collaborators. And we go in and we do this harvesting. We plant them to these migrated genotypes and many, many species, trying to create resilient forests. And then we're measuring the impacts on the carbon cycle, on the water cycle, on the biodiversity, and the regenerative capacity of the forest. So this project has been funded through basically scientific grants so far. But as we go on, it's like it's a 100-year project. Obviously, I'm not going to be alive until, in 100 years, but my students, of which I've got, a couple dozen students working on this, are going to shepherd it through into the next generations. And it's really important to do this work to demonstrate, not just to study and understand, but to demonstrate to people that there are alternative ways to do things. There are other approaches, and they work. We found that when we leave these old trees, we store carbon better. When we have a diverse community, there's way more sequestration. We have more biodiversity in the moss layer and more biodiversity in the lichen layer in the canopies. We have more small and large mammals using these forests, different communities. And so, we're demonstrating that this works, and the work is ongoing. We're always applying for money. It's really draining, but we're also getting funding from some foundations now as the word is getting out, because people are seeing it as a really positive way that they can use, they can apply this in their own forests. And it doesn't have to be a temperate forest in BC. It can be a temperate forest in the US, or it can be even a boreal forest or a tropical forest. These principles still apply. How can you get involved? Well, you can contact me and get involved, or you can also just donate funding to the project. And there is a website called mothertreeproject.org, and there's a donation button there, or you can donate to UBC. I would be just as happy if you donated to the Fairy Creek protesters or any kind of conservation movement that saves forests. But definitely ours is very much about regenerative forestry and alternatives to clear cutting to change our pathway to show people that there are better ways to do things. So that's it in a nutshell.
Yeah. And that's awesome. And I'm really glad that we had you here this evening and that lots more people heard about your awesome work and maybe get a hang of it and will donate something, or just read the book and spread your word so that, especially the young people have to know that, have to learn that there is collaboration possible if you want to, you know.
And it gives them hope too. And it gives them agency when they actually belong to, something that works. It creates so much momentum to do this kind of work.
Yeah.
Yeah.
I mean, I can only hope that it opens the eyes of many people. I mean, we had even members which questioned, the idea of a book about trees. What does this have to do with a mushroom club? And so I really hope if they listened that this opened up their eyes and got rid a little bit of this tree blindness, which we all more or less have, and get a new understanding, because we didn't grow up like First Nation people, which I often wish we could, but we are not. So we have to work on it. Mm-hmm. Anyway, I totally loved it. Thank you so much.
Well, thank you. Thank you very much. And thank you for all these wonderful questions. I'm sure you all have more questions, and, I'd love to talk to any of you. So, you can shoot me an email if you still have a question unanswered. But thank you all.
Thank you so much, Suzanne.
Thank you. Take care, everybody.
Okay.
You have a good night. Yeah.
You too. Bye-bye.
Bye. Okay, so just to wrap up, we'll cover all the info and questions about 4As and show in a email newsletter coming up. Thank you. It looked like we had a lot of folks here tonight who are either new members or just signing up for the first time or coming from as part of another club. So hopefully you'll check out our website at kitsapmushrooms.org and join us again in the future. So thanks everybody for all your great questions, and check your inboxes.
Yep.
Okay. Thank you and good night. Great job, Kirsten.
Awesome. Good job. Good job, John.
Thank you, John. Thank you, Kirsten. We all appreciate it.
All right. Good night.
All right.
And thanks for watching. Take care.